Afternoon. Today we're going to tackle questions 5C and 5D together. So 5C, it says genetic modification is carried out by gene transfer between species. So all we're doing here is we're taking DNA from one organism and we're moving it. We're transferring it to a different species. Thus, gene transfer, moving DNA from one species to another. This is the foundation of work with GMOs, and it is something that you need to be very, very comfortable with because a lot of our society does not understand how this works. So gene modification by gene transfer, right? You're changing genes by moving them from one organism to another. This is also commonly known as transgenics. Again, moving genes. So what you have is you have organism A and you have organism B, and there is some gene that is carried and expressed in organism A, and you want that expression to occur in organism B. So how do you get genes to be expressed here? You simply move them. You isolate them and then introduce them into the second organism. So gene modification leads to an organism that is genetically different. It leads to a GMO. Now, people tend to have an emotional reaction to that term there, and then you start discussing the science behind it, and a lot of people don't fully understand how GMOs work. And so when we talk about a GMO, all a GMO is a genetically modified organism. So when you start hearing about the fact that we eat GMOs and people have a response to that, you need to sit down and find out what are the genes that were transferred and why, whether these genes made the plant more drought resistant, made it grow faster, made it grow more robust, resistant to insects, resistant to fungus, possibly resistant to a compound called glyphosate, and that's where Roundup Ready crops come from. And that's a whole nother discussion because there are issues with the application of Roundup Ready crops, but there's no issue with us consuming those crops. So 5C is meant to introduce you to what a GMO is and the idea or the concept that it is simply moving a trait that you find useful in organism A, and you want that useful trait to be carried and expressed in organism B, and so you move that DNA. By moving the DNA, organism B then goes through transcription, translation, and expresses that protein, causing a certain characteristic. So that's 5C. Now we get into 5D. So 5D says clones, whole nother discussion. It says clones. Clones are groups of genetically identical organisms derived from a single original parent cell. Animals can be cloned at the embryo stage by breaking up the embryo, that's controversial, into more than one group of cells. So when people talk about cloning being unnatural, that is an inaccurate statement. Any pair of organisms or even triplets that are identical twins, identical triplets, identical quadruplets, those are clones. They are all genetically identical. When someone says that they are against cloning, what they really mean is they are against human cloning, and that's a different discussion. So we need to be sure that you are comfortable with the fact that we as a species, as other organisms do or other animals do, create clones. So, female reproductive system. Now, I'm intentionally magnifying or exaggerating the one side of her reproductive system. Of course, her oviduct doesn't really look like this. But what are we looking at here? These are the labia, vagina, cervix, uterus. This would then be her left oviduct, and there's an ovary over here. here is her uterus and here is her right oviduct. And so here is her ovary. Here is her gonad that produces ova. Cool. So then about once every 28 days, the ovum produces, the ovary produces an ovum. Process called ovulation. We will get more into that your senior year. And that ovum, if she has unprotected sex and is inseminated by a male, can then be fertilized to make a zygote. So I'm throwing out a lot of vocabulary there, so let me catch up to that. So the process of the ovary releasing an ovum is the process of ovulation. You then get a haploid sperm and a haploid ovum. These two haploid gametes join by the process of fertilization. The process of fertilization forms a diploid zygote. Done. Now that diploid zygote starts going through mitosis and goes from 1 cell to 2, and 2 cells to 4, and 4 to 8, and 8 to 16, until you have a cluster of cells. And we talked about this at the beginning of our class together back in July and August. So this process of going from 1 cell to this cluster of cells, all of this is mitosis. There's no genetic variation here at all. So this is a process of cloning. Mitosis makes genetically identical daughter cells. And so mitosis is by definition cloning, which is why when people say cloning is unnatural, that is just not an accurate statement. Mitosis is the process of cloning and forms all of the cells in your body except your sperm and ova. So we then get this cluster of cells here. These are undifferentiated cells, and this is called a blastula. Now, in some individuals, this blastula breaks apart, and you will hear people say the blastula spontaneously breaks apart. Now, you should have a reaction to that whenever you hear someone in science say, oh, that's spontaneous, that is our lingo, that's our vocabulary term for a process that we know occurs, but we don't know what causes it. We don't know what regulates it. We don't know what starts it, stops it. We simply know that this process occurs and we can document it and study it. But what causes this blastula to break apart, we don't fully understand. Thus it is spontaneous, right? So then this blastula will then break apart into 2 smaller clusters of cells. And then from there, they will start to differentiate. And this is when you get into the gastrula and gastrulation, and you start getting cell differentiation, and the bottom of the cells starts to invaginate. You get the formation of the large intestine. Fine. So this process right here leads to 2 genetically identical individuals. Since these 2 individuals are genetically identical, these are twins. If this happens again, you would have identical triplets. So this is how identical twins are formed. It's a single ovulation. It's a single fertilization event. It's a single diploid zygote. And for some reason, and we don't know why, the blastula breaks into multiple clusters of cells that are all genetically identical, and thus you get identical twins. So when you write this up in your notebook, please group questions 5C and 5D together.